This is for exercise 3B, plant cell mitosis. So you'll be setting up a drawing page and divide it into 6 equal parts. And on another video, I'll actually be making the drawing for you. And the 6 different sections of the page, the subheadings are these 6 procedures. So from left to right and top to bottom, there'll be interphase, prophase, metaphase, anaphase, telophase, and daughter cells. Those will be your 6 subheadings. And I'm going to give you a corny crutch to remember these in order. So right there on your lab menu, if you want to underline what I'm telling you, I've got a crutch for. Underline the I on interphase, the P on prophase, the MET in metaphase, the A, anaphase, the T on telophase, and the D on daughter cells. And I got a corny crutch for you. So you put this off the side somewhere, and it's how I use to keep them straight. And you may have to replay it again on any of this, but you can always stop it and replay it. Inez, pour me another tequila, dear. Inez, pour me another tequila, dear. I mean, I could have used tea, but too much tea doesn't give you double vision. Too much tequila, you'll start seeing double. And daughter cells are duplicates, or 2. So that's why it's Inez and tequila. Now here's a corny crutch to remember what these phases look like. Some are translations and some are just a corny crutch. INTER is Greek, and it always means between. Interstate, intercontinental, anything means between. So interphase is between divisions. Prophase is corny. The chromosomes look like a pile of worms. On prophase, the chromosomes look like a pile of worms. Interphase, you can't see them, just like you don't see the play or the movie during intermission. It's between acts or between movies. Prophase, they look like a pile of worms, no particular pattern to it. On metaphase, underline the MET, and the chromosomes have met right across the center of the cell. They have met. On anaphase, the A stands for they have pulled apart. The T for telophase, put tightened. They've gotten shorter. So telophase, the T, they have tightened. And this is what the chromosomes look like. On daughter cells, they have doubled, or there's duplicates. There's 2 of them. And so if you use the name, it almost tells you what the chromosomes or what the thing is done. So the crutch is, Inez, pour me another tequila, dear. And the other will actually describe what the things look like. And so I'll be making the drawings on the board later, but right now on the screen, you'll see that there's 4 cells right in the center, kind of making up a square of 4. Well, those are interphase. They're not actually undergoing mitosis, so they're in between divisions. And if you look at procedure 1, I'm going to point out what all these parts are. We'll be labeling them later on the drawing. But you'll see that they all have a border, kind of a rectangular or square border around. That outside is the cell wall. All that just kind of grainy stuff just inside there, kind of pink looking, that's cytoplasm. The large round structure in each one is the nucleus, and we'll label that with a bracket. And then the actual lining that makes up the edge of it, the border is the nuclear membrane. And all of that just purple grainy stuff inside the nucleus is chromatin network. It's actually the chromosomes, but they're so stretched out, they're not distinguishable. Now, don't draw that dark purple spot. Just don't even draw that in. That's a structure that we're not going to deal with. It's a nucleolus, but don't even draw it in. The whole big round purple thing is a nucleus with a membrane around it and the stuffy and chromatin network. Now I kind of mapped these so I won't spend a lot of time, And I drew me a little map here, and I think I can go and find these phases in order. So I'm going to go up and down and show you a prophase. Remember, a prophase, the chromosomes look like a pile of worms. So maybe it won't make you dizzy. I'll go slow here. And I'm going to go down and find a prophase, and I'll point it out when I get there, and then I'll put it in the center of the screen. And this is as large as I can make it for you. And so now you're looking for one that is about in the center of the screen, and you see it looks different from all the others around. You see white space because the chromosomes have organized, but they're not in any kind of pattern. It looks like that pile of worms that I used for my crutch. So do you see the one that looks different right in the center of the field? That it's got the dark purple spot that you don't draw or just ignore. It's round, but there's no membrane present. And if you look at procedure 2, these are the parts that you need to be able to see and identify. So in procedure 2, it has cell wall, and that's just that border around it. It also has cytoplasm, and that's just the kind of pink grainy stuff outside. And all of those purple little chain-looking things, those are the chromosomes. They're visible. If you look at the interphases all around, you don't see the distinct little chains. So that's prophase, and the chromosomes look like a pile of worms. Now that's a better one. I'm going to move up and find another one. It's not as picture perfect as that one, but hang on while I slide back up and find a different one. I'm hoping you can identify it. It's not as picture perfect as the other one, but you see the one there close to center that you can see white space in it, and there's 4 interphases right below it, and I'm kind of taking it out of order here, but do you see that there's 2 right under that prophase that are a whole lot smaller than the prophase up above it? And if you look to the cells to the left, you see they're 2 or 3 times larger. And if you look to the right, you see how much larger those cells are. So those 2 cells right below that prophase are daughter cells. They're just baby interphases, and you can see that they each have a cell wall, each have a nucleus with a nuclear membrane, and that chromatin network inside. So daughter cells are just 2 just formed interphases. That's going to be procedure 6. And you'll label all the parts that you had on interphase, but you'll put some on one of the daughter cells and some the other. I'm going to go now to metaphase, so it's interphase, prophase, and then I'm going to go over to the right and see if I can find that metaphase. Now, it's not a picture perfect one, but it's the best one I could find on this slide. And let me get it in the center here. Now the cell wall is not real distinct, it's rectangular. And I'm going to focus up and down on it because they're at different depths. And you see the kind of dark purple chains, and they're just kind of zigging and zagging across there. Now they have met in the center. They went from being real disorganized until they've lined up horizontally across the cell. Now you're going to have to draw the cell wall in. It's not real distinct, but those chromosomes there are in metaphase because they've met across the center of the cell. If you look at the label it calls for on plant cells, there's a part there that's not visible as well as it is on animal cells. So in metaphase, of course, we'll have drawn in the cell wall. All those dark purple things kind of horizontally lined up are the chromosomes, and the pink grainy stuff is cytoplasm again. And we're going to have to fake this in. When you draw the chromosomes real bold with your pencil, between at the top and the bottom, real faintly draw just some lines going to a point up above it and to a point down below it. Those are spindle figures. It's what the chromosomes are going to travel on for the next step. So that is metaphase, where the chromosomes have met. And you label cell wall, the dark purple thing's chromosomes, draw in the spindle figures going to a point above and below, and then that pink grainy stuff outside is the cytoplasm. And I'm going to anaphase. It's a pretty good one, and it's going to be to the right and down. So let me see if I can find it like I did earlier. I'm going to go to the right and then go down. Okay, here it's coming. Let me get it to the center here. Now they're kind of at an angle, and I want to focus up and down on it. Now you can see the spindle figures as I'm looking through the scope. Now the camera may not capture it as distinctly as I see it here. So you see the chromosomes have, half of them went to one end of the cell, and they migrate kind of like to two different poles. And so it looks like a bunch of V's, and they're attached at the center, and so that's anaphase. The chromosomes are pulled apart. They're all together in metaphase, and half of them go one direction and half the other. And I want to focus it up and down, and if you can see some faint, super faint little lines between them, those are the spindle figures. So draw those in very, very lightly. Don't let them compete with the dark chromosomes. And so the labels you'll have on that, and I'll be putting it on the board. The cell wall is the rectangular border. The chromosomes are those dark purple things that are kind of V-shaped. The spindle figures, you draw it very, very faintly between them, and the cytoplasm is just the pink grainy stuff outside. And then the chromosomes that you see pretty long there, they're going to tighten on telophase. So now I got to go back up and to the left for a telophase, so don't let it make you dizzy. So I'll go slowly here so it won't make you dizzy. And then go up. Okay, here are those tighten little things. Let me get it focused better. Okay, now let me move it to the center. Okay, I'm going to focus up and down on it. And the cell wall is kind of mutilated here, but the chromosomes are like picture perfect. So you can picture those others would be like if you had your hands across from each other with your fingers extended, and they're long. And then if you kind of bend your fingers down and they tighten or shorten, those dark purple things there, they're really tight, but you can tell there's individual ones, but they're just really shortened. That's telophase. And you can kind of see grainy stuff between them. Those are the spindle figures. You need to add the cytoplasm, the pink grainy stuff. And then at some point, a little line will start forming across the spindle figures, and it's the cell plate. Now this one hasn't formed the cell plate yet, but draw it like this, and then draw a faint little line between those two masses of chromosomes and label that cell plate. I have one over here that shows that, but the chromosomes aren't near as good. So this is a picture-perfect telophase, but you're just going to have to draw that cell plate. So if on the practical you see one that looks like that with a horizontal line between those two masses of chromosomes, that will be the cell plate. it continues to grow until it goes all the way across, and it'll become the dividing cell wall between two daughter cells. So you go from telophase, and I showed you the daughter cells out of sequence a minute ago. So what will happen, the chromosomes will start rounding up in a round shape. A membrane will form around them. The chromosomes will break up again to where you can't see them into specifically the chromatin network, and they're just going to be two smaller interphases. I'm going to ease back down, and here they are. You see the telophase that's up at the top, and then you have the two identical same size cells right below that telophase that are smaller than the cells up above it, to the left and to the right. And that's just two baby interphases. So that is the 6 phases of mitosis. The order is interphase, prophase, metaphase, anaphase, telophase, daughter cells. Or Inez, pour me another tequila, dear. Okay, I'm going to shut this off, but you can back it up. You can replay it. And on a practical, there'll be one of these, and they'll say, what phase is this? And there could be a pointer and said, what is the structure at the end of the pointer? So be sure you know where the cell wall, the cytoplasm, the nucleus, the nuclear membrane, the chromosomes, and all those parts that are bold-faced. So I'm going to cut this off now, and then I'll film the drawing, and you're probably going to do a better job on this blackboard or this whiteboard. I can't shade in, and so when you shade in interface, don't put any distinct lines, just shade it in, and I'll demonstrate that the best I can. So here's signing off, and go starting the other one.